You know, vaccine is very, very difficult to, very difficult to now need to know to have an efficacy vaccine now. People ask me anytime, how about efficacy vaccine? It's the right time now to learn more about that. And this person can talk to us about this. Dr. Lawrence Corey is a member of Fred Hutchinson Research Center, professor of medicine and laboratory medicine at the University of Washington, and past president and director of Fred Hutch. Dr. Corey is also principal investigator of the HIV vaccine trial report, an international collaboration of scientists and institutions dedicated to accelerating the development of HIV vaccine. His honor is awards include election to the National Academy of Medicine and the American Academy of Arts and Sciences. He is also the recipient of the American Society of Microbiology Chemists Award for Lifetime Achievement in his role in the development of antivirals for the treatment on herpes viruses and HIV. You know that this person is a very, very noble person, and we are very welcome you to be coming here and to talk to us about the vaccines. Thank you for your time.
Thank you, Professor Dea, as well as the other chairs. It's a delight to be here in Kigali and beautiful country, as you can see. So I'm going to get right to our talk today and hopefully be a little bit provocative about where we are in the HIV vaccine field. If we could maybe get to the next slide, that would be helpful. HIV is still the world's most important global health problem. That's why we're here. It is the epidemic of our time. Globally, the virus is unfortunately doing well. There are 1.4 million new infections yearly. That's 5,000 acquisitions a day. We still have an issue with mother-to-infant transmission. There's still 180,000 infants a year still infected. We now have 37 million people living with HIV, 76 million since the epidemic started. There are 770,000 HIV-related deaths in 2018 alone. And essentially more cases are being acquired than deaths per year, so the epidemic is still growing. The epidemic is also quite diverse. There are regional differences in both vulnerable populations as well as incidence. And even though we know when mother-to-infant transmission is occurring, we're also still not able to totally solve that problem. These slides, next slide, from the regional differences in incidence of HIV infection, show at least in Eastern and Southern Africa, we really are, through our prevention methods, making a difference. We've had a reduction of approximately 30% over the last 18 years. Similarly, in the Asia and the Pacific area, we see some reduction. In my own country, hardly any kind of reduction. In Eastern Europe, we see increases, marked increases, 29% increases. In Latin America, it's about stable. So overall, we're seeing some measures of success with our toolbox, but we still see major increases in incidence in many areas of the world. Next slide. Another illustration is taking these data from UNAIDS and just putting them up there by region to show that if you just look at the rings, which are the populations by region, you see that the concept of just actually having selected groups as the main vulnerable populations for HIV is just really not true on a global level. We certainly have, in many areas of the world, increasing amount of what I guess I'll call, I don't know if it's that chartreuse or green, which is sort of the general population. So we are starting to see increasing amounts of generalized populations for HIV as it spreads from commercial sex workers or men who have sex with men, transgender persons into the general population. Next slide. Probably our best reduction has been mother-to-infant transmission, but yet as you look at it in southern and eastern Africa, it's still 10% overall, and many other areas it's 17 to 20%. Next slide. So if we look at adults and children newly infected with HIV over the last, from 1990 to 2018, and you look at that green dot, which is what UNAIDS has said they want us to get to, which I don't actually think is, what I would define as an AIDS-free generation. That green dot is around 500,000 new cases still a year. To me, an AIDS-free generation is less than 100,000. We're not even close to be able to get to that number globally with our current toolbox. So my main message in this talk is that the current tailored toolbox for prevention is not getting us to where we need to go. And that's the main message. My commentary on prevention strategies should not be construed that I do not think that they should be implemented and work. Many prevention strategies have high efficacy in clinical trials, but the problem is their extended effectiveness requires continuous adherence, circumcision accepted, which often results in decreased effectiveness over time. They require high saturation. Durability and saturation are the things that have to be done on a population level for long-term effects. And whether it's condoms, or PrEPs, or vaginal rings, or PEP, they all deserve support. They all require increased uptake. But the recent studies that have come out in Pop Art, etc., say that despite the massive efforts that have occurred, we are not going to get to the area where we need to go. So we need new technologies. Test and treat is an important strategy for individual health. It can have an effect on transmission. U equals U is correct. But long-term adherence and prompt identification of HIV infection is just not translatable or scalable on a large scale. It's not even eliminated mother-to-child transmission, which has a very definable exposure. And most African countries have made huge strides in HIV treatment programs, but there's no evidence this is sufficiently reducing the risk of HIV acquisition among countries with a high density epidemic. So that's a reality. That doesn't mean we stop what we're doing, but it is a reality to say that we need more in that toolbox, that we need to develop and continue to develop new technologies. We need, in the technology terms of Silicon Valley, a disruptive technology. And that means in HIV vaccine. There is no precedent of ever controlling an asymptomatically acquired, asymptomatically transmitted, sexually transmitted infection unless we have a vaccine. That is the disruptive technology. Now, why has it been so hard to develop? I've been in this game for 20 years. I told my boss when I first started this program at the Hutch to take a decade. I was wrong. It's hard. It's a lot easier to develop antivirals than it has been for an HIV vaccine. The genetic diversity of the virus is greater than any other pathogen. The envelope is innately less immunogenic because the virus has all these sugars around it, which deflect and make distracting antibodies. And we're finding out about those distracting antibodies. We can take a rabies envelope and shove it into almost anything, and we'll get a great vaccine. You put an HIV vaccine into that same vector or shuttle vector with the DNA, etc., the immune response is about 100 times less. It is the baldest of all the viruses. The viruses usually have many envelopes on it, almost 60 or 48 on the outside of it. This one has 8 or 10 or 12. So an antibody can't grab onto it and grab onto the other side. And those antibodies, therefore, can't lock on and can't really have high affinity. The animal models are expensive and non-predictive. And we have to be clever. There are really no people, we're 0 out of 76 million of people spontaneously curing HIV. That is different than any other viral infection, whether it's hepatitis C or hepatitis B. So we don't have a model of studying the people who have self-cured. And so we have to be cleverer than the virus. And the science has gotten better and better, but it's requiring work to do this. It's also been hard to develop a vaccine because we've had to develop new structures for vaccination. Commercial companies are not, it is so high risk that they dropped out of this field in the early 1980s. So we in the nonprofit world, whether it be in the universities or in the NGOs, have had to fund this. And it has been philanthropy and mainly the US government, but starting maybe to be some other governments that have moved forward in developing the vaccine, and also for us in the public sector to develop manufacturing complicated vaccine products. This has taken a while to develop. We've now finally been at the stage. So I am saying we are making major inroads in both these scientific and policy issues. And the HIV vaccine field, you're going to hear, is moving forward with, I will say, some excitement and cautious optimism. Now, one of the first issues that's occurred is it conceptually possible to develop an HIV vaccine? And the answer is, if we have that answer, the answer is yes. The RV-144 trial, which was announced almost a decade ago, showed that you could get protection. This is a trial in Thailand of a complicated vaccine. We'll talk about it in a minute. But overall, in the first year, it had 50% protection, and after 3 years, 31% protection. Not where we wanted to be, but yet conceptually showing that you could do something. One of the important things about this trial was that it set forward an international effort to find out why some people were protected and why some were not, what we call correlates of protection. Next slide. Next slide, I'm sorry. Next slide, next slide, I'm sorry. Keep up with these. Next slide. Next slide. All right, that is the RV-144 trial. We see the separation within the curves. Next slide. Now, when we studied this trial, we found no correlation between the normal kind of antibodies that work with vaccines, which we call neutralizing antibodies. They didn't neutralize the viruses in Thailand, but there were antibodies that we call non-neutralizing antibodies or binding antibodies that are associated with vaccine efficacy. And we identified what antibodies and to what regions of the virus that those antibodies locked onto that we could then sort of duplicate and design vaccines that we hoped would be better. We call those virion binding or infected cell associated functions. Next slide. One of them is a complicated slide, but it has an easy concept. That line that goes straight up over the green is really the people, the vaccine efficacy of people who had high titers to an area of the HIV envelope to which immune pressure was put on the virus. In other words, if you make an antibody to that part of the virus's envelope, heretofore we didn't understand that. You actually got protected from this vaccine. And this antibody is not naturally made very often. And so it gave us a clue that if we made vaccines that elicit high titers of these antibodies, we are hopefully going to get better protection. So after the RV-144 trial, next slide, there was a formation of a public-private partnership between two vaccine companies that were interested in making a vaccine. One of them made a vector that was a canary pox vector. The other one that made a protein, a recombinant protein called GP120. And in concert with the Gates Foundation, National Institute of Health, South African Medical Research Council, and the U.S. Military Research Program, formed a partnership to actually manufacture a vaccine that we hoped would do better. A vaccine that would be directed at the Clade C epidemic in sub-Saharan Africa. Next slide. And a vaccine that we thought would give stronger antibody responses than what we saw in Thailand. And so we constructed a new LVAC vector. If you could hit the next slide, that will circle that. We constructed a bivalent subtype C GP120 with a new adjuvant, circle that. We decided to give booster doses because vaccine immunity waned during the Thai trial, and we hope to optimize this regimen. Next slide. We then did a series of trials. The first one was in South Africa, that was led by Professor Linda Gray, was to take the RB144 regimen and see if South Africans could actually respond to it as well as Thais. They actually did that even better. The second one was the trial in which we put this new vaccine regimen into South Africans and other people in sub-Saharan Africa. That was led by Linda Gale Becker, who's in the audience. And that showed that we could actually get better immune responses than we saw in RB144. And that led us to an efficacy trial, which we have called HBTN 702, which we started in October of 2016. And I'll describe that in the next slide. This is a trial that is now fully enrolled of 5,400 persons in South Africa. 70% are women, 30% are men. Getting this regimen in which they get many doses. Up here you'll see that there are 6 doses over an 18-month period of time. And it's a randomized controlled trial. Next slide. Just to give you a sense of what a massive undertaking of this is. These 5,400 persons were enrolled in 14 sites in South Africa. There are approximately 600 research personnel involved in the trial. We enrolled 5,477 persons that were enrolled by June of 2019. We collected over 200,000 vials of PBMCs. And you can read the bottom of this. We monitor for whether PrEP is being taken. We collected essentially almost 500,000 vials of serum, and we're looking at whether the microbiome affects whether the immunogenicity of the vaccine. And we've taken almost 8,000 swabs from the vaginal tract and the rectal tract to look at the microbiome. So it's not a trial, it is a trial to not only figure out whether it works, but how it works. And that is, we'll come back to this, the incredible, wonderful infrastructure that has been built with African scientists and collaboration that is going on in the field of HIV vaccines. Next slide. There's another program also ongoing in the African continent. Next slide. And it was a program that was initiated by Dr. Dan Baruch through the NIH funding, looking at a different approach to HIV vaccines. This one, instead of using a recombinant canarypox vector, is using a recombinant adenovirus vector that's a cold virus that carries the HIV genes in it. The genes are different. They are using synthetic biology to actually cover the genetic diversity. So they've actually been designed in a way that they're not natural strains. They're using mathematics to actually design the envelope of HIV so that that it will essentially mathematically cover the genetics of over 95% of all the strains that are circulating globally. So you take the first 9 amino acids and you put it in the things that cover the greatest concentrations for amino acids 1 to 9, and then you go 10 to 18, and you march down the entire genome of HIV, the 160,000 genome. So these strains are not found in nature. They're using mathematics and synthetic biology, and they are providing some really interesting answers. There was recombinant boost also, and this vaccine was studied extensively, next slide, in non-human primates. And in challenge models of non-human primates, it worked very well, up to 70 or 80%. Next slide. We did the studies large enough. The only thing important in the next slide is to look at the upper right-hand blue column. And we looked at the immune responses in the non-human primates who were challenged with sort of what we call SHIV, which is a recombinant virus that has an HIV on the outside. And we looked at the immune responses of the animals that were protected against experimental challenge versus did not. And we saw that the animals that had made very high responses in antibodies, again, non-neutralizing antibodies, and very high responses in their T cells, those were the animals who were protected. So we then asked the question, can we make a vaccine in humans that is very similar to this and achieve those levels of antibodies? Next slide. And so we went ahead and did that. Next slide. And the answer is yes, we could. So this vaccine, which has, again, uses an adeno-26 recombinant vector with a protein boost, was done in a series of studies, including a study that was done in Rwanda, as well as the United States. Next slide. And it showed that the immune responses were as high, if not higher, than we went into non-human primates. I just want to illustrate that this vaccine is a complicated vaccine. It's a mixture of 4 separate different adenovirus vectors that are put into a vial. 2 of them that are enveloped, and 2 of them that are the inside of the virus. And then it's boosted with a recombinant protein that in sub-Saharan Africa is a recombinant clade C GP140 protein. Next slide. And in 2017, that slide, we took this mixture of 4 mosaics and the recombinant protein and brought it into a human efficacy trial. Next slide. This slide. We call this HGTN705. And this is a trial that has been done in sub-Saharan Africa, not just only in South Africa, but in Zambia, Zimbabwe. Can we go back? One slide. Can we go back? Yeah. Botswana, Malawi, and Tanzania. And it is a slide in just women, because where's that slide? It's a study in just women. Feeling that if we can't protect women, we really don't have a vaccine that is going to be generalizable for the world. And it again involves 2,700 women that are split 50-50 between vaccine and placebo. And again, we have 23 sites in 5 countries. We have another 600 staff working on the trial. We have enrolled 2,637 women. We have collected over 100,000 blood samples. We have collected 210,000 vials of serum. We're looking at local antibodies, and again, the microbiome and the cervix. We've collected over 5,670 of those. We've also collected cervical vaginal secretions, 23,000 of those, and vaginal swabs, 5,000 of those. So these are, again, massive trials that require an enormous logistical aspect of carrying them out. Now, these 2 trials are under control of a data safety monitoring board, which actually just met last week and told us to keep going. So the trials are ongoing, and they're designed to go for 2 years. And if there is any evidence that they seem to be working, the DSMB will tell us to go on for a 3-year period of time. So we may not know much about these trials until 2021. They are fully enrolled. They are under control of the DSMB. And we are cautiously optimistic that we will see these kinds of efficacy trials. The 2 trials will be transformative for the HIV vaccine field. People often ask me, well, why 2 trials? Well, because the vaccines produce different kinds of immunity. They produce different kinds of antibodies. Some of the antibodies are the same, but enormous numbers of them are different as to where they put pressure on the virus. And it's the reason why we are actually doing so much scientific work, in which everybody who gets infected gets their virus cloned, and we look at their immunology, is because we're trying to define where are the pressure points that the human makes antibodies that puts pressure on the virus to mutate and or protects the area of that person from even getting infected in the first place. And so there are differences, and we don't need to actually go through that. The slide says where the differences are. But the magnitude and what we now call the epitope-specific responses and the breadth of those responses differ between these two vaccines. The overlap and its diversity allows us to define what non-neutralizing immune responses are correlated, and what areas of the viral envelope susceptibility to clinically effective immune pressure. Yes, I'm optimistic that we'll see something about 50 or 60%. But to really get to where we want to be like measles vaccine or rubella vaccine. That's at 80 or 90%. So if we could get a correlate out of 50 or 60%, we will reiterate that vaccine and make it better to get it from 60% to 70% to 90% to 95%. It becomes not a biology problem, it becomes an engineering problem. And frankly, with the tools that we have to engineer vaccines or almost anything in biology now, we can do that if we can make it an engineering problem. And we will answer the question whether non-neutralizing antibodies can be clinically useful. Now, the next area of HIV vaccines that I'm going to talk about is the era of neutralization. Because we finally have to develop technologies that answer the question, why do people develop neutralizing antibodies so late in disease? They've already shown you the next slide, which is the fact that antibodies have been passively administered, serum antibodies, for hundreds of years in infectious disease. No, you just keep up with me for 1 second. Just stay on that for a while. And starting in 2009, there was a technology to develop to take the antibody-producing cells out of people who are HIV infected and started to identify the 10 or 15% of those people who are HIV infected who make what we call broadly neutralizing antibodies. Antibodies which broadly neutralize cross-clade almost all the strains. Now, interestingly, those are people who actually have lots of viral load and lots of virus in them. And their own virus has mutated away, but their body has evolved a broadly neutralizing antibody that we can actually use that to prevent infection or help design a new vaccine. And that is what we are doing in this area of broadly neutralization. And over the last 10 years, we have identified probably 20 or 30 antibodies, and probably 2 or 3 new are identified each year that have this capability of interrupting viral attachment to broadly neutralization. Now, the first one of these antibodies affected the CD4 binding site. That's where the virus latches onto the CD4 cell. And this one was isolated at the Vaccine Research Center in the United States and called BRCO1. And that antibody had very good activity. Next slide. Next slide. And next slide. Against a wide strain of viruses. Next slide. The median ID50, okay, if you can go back, median ID50 was 1 microgram, and it covered about 80 or 85% of strains. Next slide. So what we decided to do was to manufacture that antibody, and in collaboration with the sister network that we work with, the HIV Prevention Trials Network, we went ahead and decided to give a study in which we would give these antibodies passively to people. And we're doing it IV, and the reason we're doing it IV is for a reason. Not that we think IV will be used generally, because we know we can make these antibodies in another way, but IV has the ability to give us a very high concentration over a period of time. It was a placebo-controlled trial in given 8 times weekly. And there have been 2 trials, one among MSN in North and South America, and the other in women in Sub-Saharan Africa. And one of the co-chairs in Sub-Saharan Africa, Dr. Bratsu, is right here. They want me to hurry through this a little bit, so I'm going to just say that the trial is fully enrolled. Next slide. And next slide. We've had amazing adherence. And we will actually be finishing this trial in the summer of 2020. And we will be announcing the results at the R4P meeting in South Africa in the fall of this year. One of the reasons we're excited about these antibodies is that we know, next slide, we can make better antibodies with very long half-lives. We could actually give these antibodies, if they worked, subcutaneously by self-injection or by a hospital injection, and they will last 4 to 6 months. Next slide. We also know that we can give combinations of these antibodies. And we can make antibodies that are just like antiretrovirals, are at different parts, block the virus at different parts, not just at the CD4 binding site, but at other parts of the virus. We can put them into combinations. And we know those combinations are more effective. And so, even though we've given this first as one antibody, we know we can make 2 and 3 antibodies. And we know, once we define what the level of neutralization is, how to make a combination that will actually get us to very high efficacy. Next slide. We can have the next slide. The next slide. Next slide. So I'm going to end on the following 2 notes. If any of the HIV vaccines or any of the antibodies in efficacy trial testing are effective, it will, in my opinion, unleash an enormous explosion in scientific inquiry to improve, adapt, and most importantly, bring to the world a new form of HIV prevention. Yes, an HIV vaccine will be the most complex vaccine ever designed. It's not going to be a single thing. It will be multiple injections with multiple different vaccines. It will be an implementation challenge. It may even disrupt the way we diagnose HIV because it will give antibodies to HIV. But they will overcome the current barriers to population-based control of HIV and provide a tool that could get us to an HIV-free generation, a reality that is not present with our current tools. And yes, the science behind such a vaccine will have additional spin-offs. Next slide. Vaccine science is well established on the African continent. There has been a massive investment in HIV vaccine science and infrastructure that has been built on the African continent in the last 7 years. HV10 alone has 54 clinical trial sites in Africa. We have built a sophisticated immunology lab in South Africa, and we also collaborate with scientists all over, both in Eastern Africa and Sub-Saharan Africa, on the virology and the diagnosis of HIV infection. Every viral isolate in these studies is actually sequenced. They are actually then pseudotyped, and their susceptibility to the antibodies are being made by African scientists. Next slide. African scientists are doing B-cell cloning and monoclonal antibody isolation from patients with HIV in Africa. Broadly neutralizing antibodies are being isolated by scientists in Africa and being adapted for clinical use. Yes, the epidemic of our times continues, but the scientific accomplishments to rein it in are also increasing. There is much to do and much to accomplish if we are to get to a real AIDS-free generation. And I frankly am optimistic that it is both the antibodies and the vaccines that are going to get us to that goal. Lastly, I'm going to give a plug for my colleagues working in the area of HIV cure, who wanted me to mix this up in the lecture, but I felt vaccines needed its own forum here. I'm going to be patently advertising that there is a symposium on HIV cure in Africa that is later on this evening, and hope all of you will attend. I have many, many people to thank. First of all, the study staff, the community engagement teams, and most of all, the participants who joined the journey in helping develop a vaccine altruism is alive and well throughout the world, and I'm incredibly appreciative of that. Next slide. There are hundreds of people, these are some, next slide, people in the United States. These are the people who fund us, next slide. But our real point here is our collaborators in Africa. There are too many to list. There are many in this room. I have the privilege of working with an incredible group of people, of African scientists, who are really accomplished. And I have great pleasure. The great pleasure of my job is interacting with them. Next slide. That's the same in South America, Europe, throughout the world. Quality of work is amazing. Next slide. Next slide. My people in the HBTN. Our funders, the National Institute of Health, the Bill and Melinda Gates Foundation. Our collaborators, Novartis, GSK. Next slide. Johnson & Johnson. And the Gates Foundation and the Rating Institute. So I think that's the last slide. Let me thank you all for listening.